computer.
Okay, thank you.
Yeah.
All right. I'll somehow lose. This is nice. Weird.
I can go in and tap if you, okay.
Okay, it seems to be working. I don't know what's going on with that. It's like losing. Okay, so the big picture sort of overarching theme of the talk that I'm going to give you today is about this idea of parallel evolution. So parallel evolution is the case where the same sort of organism level phenotype or trait evolves repeatedly from a similar common ancestral state. So for those who aren't familiar with reading the phylogenetic tree, this is sort of a cartoon phylogenetic tree. It's a branching process that shows, so the blue circles there are ancestral states. They represent like a population at that time when the branching between species occurred. So this is a species tree, so we're looking at the relationship between species 1 and species 2 and so forth. We usually do what's called rooting the tree using an outgroup species. So this is something that we know is more distantly related than everything else in the tree, and that allows you to sort of put the tree in the right order. So we can say that species 1 and species 2 are more closely related to each other than either is to species 3. And we call, when they're like that, when they're the most closely related to each other, we call them a sister pair. So they're sister to one another. So the idea here is that as this clade of this group of species has diversified and evolved, there's been this reemergence of this red phenotype A 3 independent times from a blue ancestral state. So we had the blue state, and then we had something happen along the lineage that leads to species 1 and species 3 and species 5 that gave rise to this new red phenotype. And those are just things that are happening in parallel, hence we call it parallel evolution. There's a similar concept of convergent evolution, and it's like a whole philosophical bag of worms that isn't worth going into. But the point is, like, it's happening, it's converging on the same phenotype, it's happening in parallel. So one of the things that, because I have this interest in linking across scales of biological organization, is you can think about this at different scales. So for example, you can say, there's a phenotype, a red flowered phenotype, and plants exhibit that. And here it would appear to be a case where there was a blue ancestral phenotype or bluish purple, and then multiple times we saw the emergence of this red flowered phenotype. But we can dig deeper and we can ask, okay, so those are the same at like this level. If I look at it and it's a red flower, are they the same at the molecular level? So did the genetic changes that happened that gave rise to red flowers, did that happen in the same pathway? So for example, the metabolic pathway that makes colorful pigments. If that's the case, did it happen in the same protein? So this is a zoomed in 3-dimensional structure of the active site of this protein DFR. And we've actually found in another separate study in a separate group of plants, an instance of convergent evolution of the single amino acid residue changing probably the specificity in the same way. So, you can think of it, and depending on how you think of it at across different scales of organization, you might actually sort of get a different answer. Like it may be parallel at one scale, it may not be parallel at another scale. And from that, we can learn a lot. We can learn about the different mechanisms at the molecular level that can give rise to these changes in traits. We can also learn about things like constraints. So maybe, for example, there are certain things that you just cannot do in this pathway, or you cannot do in this protein for physical reasons or chemical reasons, or maybe even for ecological evolutionary reasons. Like you may have a state that's maladaptive, it doesn't work well. So anyway, by studying these cases, we can learn a lot more than we can necessarily learn from just a single instance of the evolution of this phenotype, because we can learn about things that are processes that are sort of general, that can be understood across multiple different replicated instances of this event happening. So one of my favorite examples of this is actually in birds. And so there's this great case. This is from the work of Jay Storr's lab at University of Nebraska, Lincoln. And so on the left is a phylogeny, like I showed you before, showing how all the bird species are related. And on the right, there are 3 proteins. There's the 2 alpha globins and this beta globin, and they form like a hemoglobin complex. So that's the protein, that's in your blood, and it's finds oxygen and then releases it elsewhere. And so this is a case where they looked at repeated evolutionary adaptation to higher elevation. So as the birds move from a lower elevation ancestor up in higher elevation, the hemoglobin changes its activity in order to better deal with the oxygen concentration at higher elevation, which is thinner, as you probably know, living in Colorado when you hike up, a 14 or whatever. So this then shows like all the different sites that contributed to actually changing the activity of the protein. So if it was always the same one, you just expect lines that run down where it's like always one amino acid change. Instead, it's just kind of all over the place, right? There are a lot of different ways to achieve this. And so this is like a great example to me of even at the level of a single protein, then when you go and look at the individual amino acids, you see there's quite a bit of different ways to do things. So there's like this sort of degeneracy or redundancy in the way that you can achieve the same effect. And so that's the kind of stuff that I just love and fascinates me. And I think it's one of the, sort of best tools we have for understanding general features of evolutionary mechanisms across scales. So I'm going to pivot now. I'm going to sort of tell you about the general study system in which I'm working right now. And this is the genus Penstemon. You may be familiar with it. It's a big genus in the family Plantagenaceae. There are about 280 species. Most of them are in North America. So there are a lot in the western US, for example, in the Southwest deserts in Colorado and Utah, California. There are also quite a few different species in Mexico. There's one, I believe, in Guatemala. So like as you get further towards Central America, the diversity tapers off. And then of course, even in Canada has some. So it's really cool. So they're kind of, the reason they're called Penstemon, like 5 stamen, is because they have actually 4 stamens that have functional anthers, and then they have this thing called a staminode, which is this little like fuzzy thing, and that gives rise to the common name for these, which one of the common names is a beard tongue, because it looks like it has this hairy bearded tongue. And that thing, this is a zoomed in picture from another individual of this same species in the same population, actually, that I took from. This is by west of Walden in the Park Range. And you can see the little hairs. They're like these little hairy structures that grow off of it. And they vary in color and they vary in shape, and the staminodes vary in how hairy and what parts of them are hairy. And those are some important characters for differentiating between some of the species that are more similar to each other. So like this species, you could never really confuse with any other Penstemon species because it's just a super zany one. And I love it for that reason. But there are others where you'd like squint and you could look all you want, and without getting out your loop, you would not be able to really clearly tell the difference. In the upper right-hand corner, so I'm just going to nerd out about Penstemon for a second, just because I think it's a really cool genus. But coming from the background that I came from, I didn't know that much about botany. And so I had naively assumed that nectar production, nectaries, was something that was fairly similar across a lot of different plant families or genera. And it turns out not really the case. So like when I worked before in the petunia, they have they have a way of making a nectary, or the tissue that produces nectar in the flower. Penstemon has a completely different thing. So this is yet another case of like convergent evolution on actually a pretty like massive scale. So they have these little, what are essentially like forests of glands that will appear on 2 out of the 4 functional stamen filaments right at the base, and they just sort of secrete nectar that pools up in that part of the flower. This is anthocyanin pigments, which I'll talk about a bit more, that give it this lovely color that are in petal cells. So this is peeling off a single layer of petal cells, looking at it under the microscope. And then in the bottom are the actual anthers at the end there. And that can also be a very diagnostic character between certain species. So to just continue rambling about Penstemon, we have a bunch of them in Colorado, actually. So as members of the Native Plant Society, you've probably seen them, or if not, you should go look for them. These are some of my favorite ones. This is probably maybe my favorite one. We have Penstemon hollyi. It only grows up at really high elevation, like 11,000 plus feet in alpine tundra, and it doesn't flower for all that long. It has like tiny little vegetative tissue part and huge fat flower. So it's like kind of a weird combination. There are other ones with really tiny flowers, like Penstemon reverbii in the upper corner. This is a Penstemon Whipple-anus, again, the one that I showed you before, and it has this very distinctive polymorphism between a white and a purple that often are together in the same population. And there's actually there's a graduate student in Carrie's lab, my colleague Zach, and he is working on that. And he's actually found my iNaturalist data to find interesting populations has been super useful to him. So, if you're out looking around, take pictures, put them on iNaturalist. I really encourage you to do that because it is very helpful. Okay, so back to the sort of the meat of this is what are the molecular mechanisms, the genetic mechanisms that underlie this sort of these repeated transitions to the same hummingbird pollinated phenotype. So that tree I showed you was actually from Penstemon, as you might have guessed, and that is a subgroup within Penstemon called section Habroanthus. And Habroanthus is total about 50 species out of the 280 species. So it's a pretty good chunk of the species. And within this, most of them have this what's called bee syndrome, bee pollination syndrome. So they tend to have sort of shorter, wider, blue-purple flowers. They have shorter stamens and styles, and they make pretty little nectar. Hummingbird pollinated ones, they're kind of defined by this set of really characteristic traits. So they make a bunch of nectar. The stamens and the styles are long because they have to stick out to the end of the flower and they have to touch the hummingbird's head. And then they're long and narrow and red, so they're sort of well adapted for the hummingbird to fit its beak inside. And red is thought to be, it's a common, especially in North America anyway, it's a very common adaptation to hummingbird pollination because the birds can see red really well. So that has oftentimes the color has a lot to do with which pollinators you're attracting. And these different components, the phenotype, have to do with what aspect, you have to be able to attract the bird, you have to be able to feed the bird well enough that it wants to come back. There's all these sort of functional considerations that have evolved this amazing matching. So, for what is essentially a random mutational walk, it's pretty amazing how these things just happen over and over again. But how do they happen? So, my current postdoctoral advisor, Carrie Wessinger, she did an experiment that's called quantitative trait loci mapping analysis. So, but basically, like it sounds more complicated than it really is. Basically, what you do is you take two species, so in this case, bee pollinated species called Penstemon brigatus and a hummingbird pollinated species called Penstemon barbatus, and you cross them, and then you do another cross. Basically, you get this population called mapping population. The reason it's called that is because it allows you to map regions of the genome that are tightly associated with the phenotype that you're interested in. So what regions stick out as being strongly associated with the changes in the phenotype that correspond to the hummingbird syndrome? And she found sort of three major QTLs, quantitative trait loci, for flower color, nectar volume, and flower width. Those are kind of some of the most diagnostic traits, and mapped where they are on specific chromosomes in the genome. And actually, these are relatively broad regions with quite a few genes in them. For the flower color locus, she was actually able to determine exactly which gene is responsible, and it's like one of the usual suspects for transitions from blue to red. It pops up a lot. I'll talk more about that later. Anyway, so we have this as sort of a backbone to this project. This is one of the pairs out of 4 in Habroanthus, and we know basically which parts of the genome at least are associated with the transition. So it's a way of sort of constantly going back and doing a sanity check for some of the other things that we're interested in asking. So first, I need to take an aside though and talk about phylogeny again. And so there's this probably arguably one of the most classic cases of parallel evolution in pollination is a case of the genus Mimulus. And so there's a famous, this tree was long thought to represent an accurate depiction of the relationships between species in this part of the Mimulus group, where M. Mimulus cardinalis, you can see sort of clearly seems to have re-evolved independently of these other red species. It's most closely related to several bee pollinated species, so it had this parallel event where you got a really similar hummingbird pollinated phenotype. And that stood, for quite a long time, and these analyses were done with a relatively small number of genomic regions. because they largely predate the era in which we can just sequence entire genomes, which now we can do easily and pretty cheaply. So when somebody did that in 2021, there's actually a guy I coincidentally went to grad school with who was just a really good scientist. So I was super excited when I saw this paper, but anyway, they did an analysis where they used about 8,000 genomic regions, and they basically you infer a tree for each gene or each region, and then you use those to collectively infer an overall tree that tells you how the species are related. And what they found was that actually it was very strongly supported that Mimulus cardinalis is actually in the same subgroup with the other red flowered species. So this actually suggests that it wasn't a case of parallel evolution. It just happened once, and then these diversified subsequently into several different hummingbird pollinated species. And so this is why I say it's a cautionary tale. If you don't know the tree, if you don't confidently know the tree, you can't necessarily even infer that you're looking at parallel evolution, let alone try to understand the mechanisms. So you may be asking yourself, how can a gene tree, how can the tree for a single gene differ from the species tree? And I mean, I remember when I first learned this and I found it unintuitive and confusing. It's, you might think like, okay, all the genes all the genes in your genome should show the same relationship between you and a chimpanzee, right? But that's not necessarily the case, because genes are not all completely linked together. They actually can have their own independent evolution, semi-independent evolutionary history. So the first way that you can get a deviation between a tree of a single gene, so that's what I'm showing on the right, and the species tree is this process that I'm going to walk through. So I'm showing here on the left the species tree, showing how the species P1, P2, and P3 are related to each other. And let's just assume we know that that's the correct topology that shows how the species are related. In this ancestral state, represented by this purple and orange circle, this is a population, and that's why I'm showing it like this sort of tube. And I think that's the thing why it sometimes seems counterintuitive. There's not just a single genome, just like in the human population, just like in this room, right? We have genetic variation at every site in our genome. Potentially, we have many differences between us. And that is the case with any population, right? I mean, unless its population is just like extremely inbred for some reason, which can happen. But so in this ancestral state, there was both an orange and a purple version of this gene. And those are, just placeholders just to keep track of which one is which. So after this speciation event that gave rise to P1 and P2 branch, or the branch that relates everything over there to P3, P3 retains the orange copy just by chance, maybe because there was actually a fairly small population. That's the only one that happened to move into that following speciation. The purple, meanwhile, and orange are both retained all the way along the way before, leading up to the point where P1 and P2 split apart from each other. And so in that case, then now we have an event where P2 now keeps orange and P1 gets purple. And so if we just look at the relationship between the genes, it's going to look like that on the right. It's going to show us the wrong relationship between P1 and P2. It's the right relationship between the genes, but it's the wrong relationship between the species. So that's this is just something you expect to always be happening in the background of all evolutionary events. Every time you're having this, looking at a tree, you expect that this is going to be happening, and it's going to be a source of deviation, of disagreement between gene trees and the species tree. The other way that this can happen is introgression. So this is the result of hybridization. In bacteria, you can also have horizontal gene transfer or something, but we don't really do that. Plants don't really do that so much. So the way that you get it is effectively the same sort of process, though. You have instead, so in the ancestor, in this case, we just had one thing, actually. Let's say this is a small population, and at this gene, they only have the purple version. So eventually, you get these things, and every lineage gets a purple version. P3 ends up with the purple version, but at the base of the P2 lineage, there's a mutation that gives rise to the orange version. And then subsequently, that hybridizes with the P1 lineage, which at that time has the purple version, and then loses the purple version and you get the orange version. And in that case, you also get discordance between the gene tree and the species tree. So these are sort of like the two big ways that this can happen. And it seems like a nuisance and just seems like, oh, it's an annoying complexity of how evolution works in the real world, but it can also be useful for statistical ways of looking for signatures of introgression, which is what we want to know if we want to study whether that's happened. And so in fact, in the case of Mimulus, what that paper went on to find is that there was very strong evidence that there had been multiple introgression events. So where lineages hybridize with other lineages, and that's part of the reason that the tree looks so kind of mixed up and was hard to infer without doing a huge amount of whole genome sequencing. And in their case, it made it look like there was a case of parallel evolution. Potentially, you could actually have it go the other way. So what does this mean for Habroanthus, Penstemon section Habroanthus? So basically, there are a few possible scenarios that I'm going to highlight. They're more like classes of scenarios because there are sort of different specific versions you could imagine of this. But one, for example, is the Mimulus scenario. So instead of the tree that I originally showed you, maybe we're just getting the tree wrong. Maybe what really happened, everything was blue, everything was bee pollinated up to one point where you got this hummingbird pollination. And then that diversified into a clade, and due to maybe subsequent introgression with bee pollinated species, it has dragged these branches apart and made it look like a case of parallel evolution when it isn't. There's a scenario where it really is just exactly what it looks like. There's just 4 independent acquisitions of the hummingbird pollination phenotype. And then, there's this type of scenario where maybe it evolved once along the lineage leading to one red hummingbird pollinated species. And then due to hybridization and introgression, those genes were introduced into other lineages and giving rise to this sort of semi-replicated evolution. So it's still an evolutionary process introducing it into another lineage, but it isn't completely different mutations that have just arisen independently. They're non-independent events. And you could imagine some subset, like maybe Cardinalis here and Eaton show this pattern, but not the other two. So, this isn't the only possible instance of this type of thing that we could see. So the question is, well, what actually happens and how can we figure that out? So again, going back to reiterate what I said before, you need to know pretty confidently what the tree looks like of the species and how they're related. So I took an approach where I used basically 5 different approaches. And the approaches vary in how I'm divvying up the genome. So how I'm sort of divvying up into chunks in order to infer individual trees for different loci, and the type of statistical evolutionary model that I'm using to infer the tree. And all of those ways, which are basically a way of sort of summarizing over uncertainty that could be caused by just the methods that you're using, they all give exactly the same pattern. And that pattern is that, in fact, it looks like we have 4 independent cases of evolution of hummingbird syndrome. So there's the Cardinalis lineage, Labrosis, Barbatus, and Eatony. And each of those, as you can see, is sort of like in the cartoon I showed, paired with blue, I've had them highlighted in blue, bee pollinated sister species. And all of these have pretty much 100% support on the nodes. So I didn't put supports on there. Interestingly though, as you might expect, because given what I said about incomplete lineage sorting, there are most of the gene trees actually don't exactly match the species tree. So there are some that do, but most of them don't. And that's not surprising. And that's why these type of approaches that integrate over thousands. So depending on the method, I'm using anywhere from 6,100 to 26,000 independent gene trees to infer this species tree. And that allows you to integrate over all of these different gene trees and get sort of a mean answer that gives you a good answer as to how the species themselves are overall related. So I need to sort of lay out the general logic for the type of statistical analysis that we can use to look for signatures of introgression. So again, we have incomplete lineage sorting and introgression as possible ways that could cause deviations between the gene trees and the species trees. And And so on the right, I'm showing a case where we want to ask the question, does the red P2 and red P3, do they show signs of introgression? And P2, we know from our species tree, has this blue P1 sister. And then O is again the outgroup. So this is just like another Penstemon, not even in the same clade, that we like put there to just make sure that the tree is in the right order. And there are two patterns at any given site in the genome that we are interested in looking at. One is called ABBA and the other is called BABA. And this is the a classic. This is the ABBA-BABA test, or the Patterson's DE statistic test. But everybody says ABBA-BABA because that's so much more fun. And the reason it's called that is because if you call them A and B, you get P1 has A, and then P2 and P3 have B, and then A. So ABBA. BABA shows that P1 and P3 are more similar, so you get BABA. And basically, what you're doing is sort of just looking at the weighting between the two. So like the ratio of the ABBA and the BABA. There's a bit more to it. It's scaled so that it lies between a certain range, but that's not really important. Basically, you just want to know, are these even or are they not? And the reason is because in the case of incomplete lineage sorting, there's nothing going on there that should make them uneven. They should come out to, like, the ratio should be 1, basically, right? They should come out to be that there's the same amount of ABBA as the same amount of BABA, and just because there's nothing else happening that would make it deviate from that. Whereas in the case of introgression, there's definitely something happening that would make it deviate from that. And so anytime you see a strong deviation from the null expectation of BABA, basically, of ABBA and BABA being equal, you see, say, an elevated level of ABBA showing that P2 and P3 are more closely related, then that's a pretty good indication that there could be a signal of introgression. And so this is like the most basic of these statistics. There's kind of a whole family of these that are more elaborate versions. And I will talk about some of those and not go into great detail, but basically all of them are based on this logic. They're just sort of rescaled or changed in a way to make it so that the assumptions match the specific question you're asking. So you're not just violating the assumptions of your statistical test, because then that's not a useful test. Okay, so one of those is called F branch, and still the logic is basically the same. We don't even need to talk about like how you calculate F branch. The idea is, if you look at this heat map and the color bar on the right, the hotter red or darker red, the stronger the signal of introgression. And this is able to actually account for the structure of the tree and do some things that allows you to actually infer, like maybe there was introgression at the ancestral node of a subgroup. And so the big takeaway here is, well, there are two. First, there actually are some pretty hot red sites in here, but none of them are hummingbird introgression events. They're all either basically B or hummingbird B. And so basically we can take away from this that there's definitely evidence that introgression is happening. And I'll talk more about this later. We know for a fact that even in real time, right at this moment, introgression is happening between some hummingbird and bee pollinated species, and between some bee and other bee pollinated species. But these asterisks highlight the comparisons that we care about for this specific question. And you can see that like most of them are white. For this Barbados group here, there is a little bit of signal in one of our Barbados individuals that we have, and like none in the other. And for anything that's contributed to the hummingbird phenotype, we should see consistent signal across any Barbados that we look at, because they all have the same red long tubular flowers. Okay. So there's a little bit more we can work with here, and that is because we have multiple different pairs. So for based on our species tree, for every hummingbird pollinated species, it has a sister bee pollinated species. For example, Barbados is sister to Virgatus, and then we can ask, for example, with this example, we're asking about the signal of introgression between Barbados and Labrosis. We can do what's sort of a mirror image, or I call them symmetric sister pairs, because we can also look at the bee pollinated sister of Labrosis and compare. And so any real signal that we have these should be somewhat consistent with each other, because that would indicate things that show that relationship to the exclusion of the other species. If that's not the case, then that is not really as strong of evidence as we would like to see to have any kind of confidence in saying that there is introgression. So anyway, I just wanted to kind of lay out that logic. So here I'm highlighting the Brigadus barbatus labrosus, Putus labrosus barbatus. One is blue and one is orange. And this is a statistic called DF. Again, this is just like that ABBA thing, essentially, but it's reformulated in a way that makes it statistically sound to use it in little chunks of the genome. So you can slide along the genome, you can take little windows, you can calculate the statistic. If you just do the standard ABBA, that like totally violates the assumptions of that model, so it's a useless statistic, but this one works. And so what you would think is that these two sort of mirror image pairs should actually be fairly, should be correlated anyway. And you can kind of just see from looking at it, and this is chromosome 4, looking at this barbatus labrosus, just as an example, you can kind of see just from looking at it that actually they're pretty not coupled. And I like formally calculated the correlations for all of these, and they're really not correlated negatively or positively. It's very weak. So I think, that says there are some peaks here, but that doesn't speak to like a kind of coherent signal of evolution between the hummingbird lineages. And you can repeat this for all the chromosomes, and they all look pretty similar. So the next thing is, we can take a step back and we can say, cool, the D statistics and everything, they're good and all, but maybe we'll just take a simpler approach. We can look just directly at the gene trees. So this is, like I mentioned, depending on the approach I used, either a chunk the genome up into windows that are like 10,000 base pairs or 50,000 base pairs, or look at actual coding gene regions. You can ask whether any of those individual gene trees support putting, like on the left, for example, all of the hummingbird species together, or 2 of the hummingbird species together. On the right, you can ask, there's an alternative, is in that case, none of them support any pairing of hummingbird pollinated species. So I did that, and this is an example, again, using the barbatus labrosus comparison. And the reason is because this is actually the highest signal we get. You'll notice this is like % of trees that support that. And, like that's 5%, so it's very low. So there's very few trees that strongly support this. And actually, the interesting thing is that for the most part, you see that the relationships between the you call it sister species of the hummingbird pollinated species actually show a stronger signal. And so that suggests that possibly there may have been introgression in the ancestor of the different clades that contain the hummingbird pollinated species. And that is consistent with what we saw in that heat map figure of the F range statistic that I showed you as well. But again, there's really no strong evidence at all that there is sort of direct hummingbird species, hummingbird pollinated species introgression that excludes the other species that are closely related to them, which is what you would expect if you had genetic changes that were directly associated with the hummingbird pollinated phenotype. So this is just a summary of all the possible comparisons that we're interested in. You can kind of see like, yeah, it tops out around maybe 2% of trees. And also it's worth noting, those two are just the most closely related in the species tree anyway. So you would expect that signal might be slightly elevated just because they're closest together in the tree. Okay, so the last thing, the last ditch effort, cross our T's and dot our I's and make sure we're not like missing some signature of this signal and getting the wrong answer. Because you don't want to confidently go, oh yeah, there's definitely no signal of introgression, and then turns out you just didn't look hard enough. So I've really been digging into this, trying to find any real signal that this is the case. So the last thing is to go back to these quantitative trait loci. So for the flower color, nectar volume, the flower width, and ask if you say go to the peak and you take a chunk around there, do those regions, which we know contribute to the phenotype, do those show relationships that put the hummingbird species, any pair of the hummingbird species together? Because that would be potentially indicative of a case where those genes or those regions were introduced and giving rise to the phenotype in another lineage. So I looked at all three. So this is the flower color locus that contains this gene F3'5'H, which we know is actually, in this case, is actually like the causative gene that's resulting in the phenotypic change in flower color. And you can basically see just that they're all still separate from each other. And they're all still sister to the same bee pollinated sister species that they always are in the species tree. That holds true for all three. So this is the nectar volume locus. One interesting thing here is that if you see that this lobrosis clade, the one that contains one of the hummingbird pollinated species, it's still sister to the same bee pollinated species, but these have moved so that the whole clades are together. And that's consistent with the gene tree thing, and that's consistent with the F branch statistics, that maybe there was an introgression that happened between the ancestors of these two clades. And that's cool and super interesting, but also doesn't really bear too much on this specific question of whether or not adaptive introgression of hummingbird phenotype was the result of whether that was the result of introgression. And the tree looks very similar for the third QTL as well. They're all still separate and they all still maintain their internal structure being sister to the same bee pollinated species. So I think to summarize this sort of evidence we have, I am fairly confident in saying, well, I'm very confident in saying these are 4 independent events of some sort, or at least pseudo-independent events. They're not all the same subgroup of the tree. Like the hummingbird pollinated species are not all sister to each other. They're not all the most closely related. And that's, consistent with this sort of one of our various hypotheses that I presented earlier, is that these are just 4 independent things. And we really do not see any evidence that that is the result of introgression. And so this, in contrast to the Mimulus case, for example, where it turned out to be sort of an artifact of not inferring the tree correctly, this actually suggests that this is sort of the hallmark example of this parallel evolution that they're really interested in studying. And so that's kind of really just the first step, establishing that that actually seems to be what happens. So I will just talk a little bit sort of about what I'm planning to do next, what I'm working on now, and how that kind of stems from this. So the kind of natural next question is, okay, so it's 4 events that happened independently. We know something about the mechanisms in one lineage, but what happens in the other ones? Are the changes at the molecular level parallel? Are they in the same pathways? Are they in the same genes, same protein? Are there, for example, are there multiple different mechanisms how you can get this long tubular floral shape? Are there different regulatory factors that you can change that cause it to develop that way? And are there, various different genes that we can change to sort of tune the knobs on the nectar volume? So I'm going to focus a little bit just on flower color, because this is probably overall the most well understood of these pathways. And so this is just a sort of cartoon schematic of the anthocyanin pathway, which produces the colorful pigments. So there's each labeled compound. So for example, chalcone is a chemical compound, and it's acted on by an enzyme, which are the little things surrounded by boxes, and that transforms it into another compound. And you get this sort of, what is essentially, you could think of as like a series of pipes with valves on them, where you're controlling the flow of material through this chemical pathway. And eventually you're giving rise to these red, sort of purple or blue compounds that mainly differ. They really just differ in how many of these hydroxyl OH groups are attached on that ring. And that is enough to change the electronic structure of the chemical enough that it shifts what light is being absorbed. So there are different places you could mutate along the pathway to change that flux. So for example, this enzyme here is necessary to get delphinidin. If you were to cut that off, you would force things to go a different path. So it's really, you can think of it like that. And I've actually not going to talk about this at all, but I've done some modeling of this using mathematical models. That was kind of an interesting exercise in theoretical stuff. So one of the tools aside from doing genetic crosses that we have that, you know, say in the greenhouse is to go look for genetic crosses that are actually happening in nature. And so Carrie became aware a few years ago of this site in southern Utah, like near the Arizona border, where there's this Penstemon laevis, which is one of our sister bee pairs, and Eaton. And this laevis is sort of embedded in this big population of Eaton. And at that site, you're getting very obvious hybridization. And there's a ton of segregating phenotypic variation. So these are all hybrids of these two parent species. And so there's differences in floral shape, and there's differences in color, and there's differences in even the plant growth form as well. So this is like a super rich, essentially a natural experiment where this is happening in real life. It's out there happening right now, and we can use it as a way to understand the mechanisms that are going on in this other pair of species. So this is a project that I've recently started working on with, and it's been well established by the other people that are on this preprint. So if you want to read more about this, you can go read that, just came out recently. Josh Stevens is a graduate student. This is mainly his project, and I've been sort of helping out with the multivariate statistics side of things, and then I'm kind of taking part of it in another direction. So this is a plot showing sort of a characterization of that segregating phenotypic variation, specifically in flower color. And so on the left, these are reflectance spectra. So basically you're just asking, you shine a light of the same light on all these flowers at the same angle and everything, and you measure what light bounces back. And that is directly related to the color that your eye sees, right? Because that's the light that's coming in, hitting your receptors. And so, you know, you can clearly see that, you know, there is a lot of color variation in these flowers, and that gets translated into the reflectance. We can use an approach called principal components analysis. That basically is a way of sort of taking this complex multi-dimensional data set. So we have measurements of the amounts of the pigments in each of the flowers, as well as this reflectance data, and sort of projecting into a space where you can actually look at it. And so what that does is just tries to find axes that are perpendicular to each other that explain as much of the variation in the data set as possible. And you can see that there are sort of groups of things. So for example, these are very red, and they make the red color gonaded pigments. And then these sort of range from a more purple blue to a more pinkish purple, and that varies in the amount of delphinidin or cyanidin, the blue or the magenta colored compound. And then those things are actually associated with properties of the reflectance. And so looking at this, we thought, okay, that means we should actually be able to, this contains a lot of information. The reflectance spectra contain a lot of information about what pigments are being made. So to test that hypothesis, I fit a model that basically you feed it a spectrum, and it decomposes that spectrum into a bunch of individual wavelengths, and then it fits this what is essentially like a really complicated linear regression model that gives you the relationship between each of those wavelengths and the pigments that's being produced. So we actually are predicting the pigments, and this is just a diagram of those coefficients. So some of them are negative, some of them are positive, and if you can see that those sort of line up with particular parts of the spectra. And so that really strongly supports this idea that there's a huge amount of information just from basically the visible color about what pigments are being made. And we don't need to dwell on this, but basically this is just testing the accuracy of the model using a bunch of iterations. And you can see, like, for example, for the red pelargonidin pigment, we have like more than 95% accuracy with this model. The lowest we get is looking at predicting the total amount, but that's still somewhere between 0.65 and 0.7. So it's doing like a pretty good job. That's like, 65% accuracy just from looking at the color. So in a way, that's pretty surprising. It was surprising to me. But the other thing that this suggests is that because this phenotypic, this segregating variation in the population corresponds so well between the molecular level at what pigments are being made and the visible color, that should also probably translate to what's going on with the genetics too, because the genes control what the pathway enzymes are doing, and those in turn make the pigments, and those clearly have a huge amount of information about the color. So what we've been doing now is doing whole genome sequencing of a bunch of these hybrids. So the parents. So we have 263 parent and hybrid individuals. Most of them are hybrids. And then we can map those sequencing reads to the genome that Carrie has assembled of Penstemon barbatus, which is one of our other Habroanthus hummingbird pollinated species. And that allows us to then look directly at what genetic variants are associated with the distribution of phenotype. So color, for example, or floral shape, or whatever else we want to look at. Interesting. So just to touch on this, we just got a bunch of this data back last week, and I've been doing some sort of preliminary analyses. So this is looking at using a program called admixture that basically just estimates what proportion of the genome came from one parent, so Eatney versus the other, Laevis. And you can see, like, here's the guys that are all Eatney, and there's the ones that are all Laevis, and there's this almost sort of nice, like, linear gradient in this population going from being essentially entirely like Eatney to entirely like Laevis. And those probably just have a few loci that are directly related to the phenotype that are in those individuals. And then there's another way we can look at this is to take that same sort of input data, and these are just all of the variants in the genome that differ between the individuals. And we can transform those again using a PCA and sort of look at how they lay out, how they are related to each other. And you can see, so the darker red and the darker blue are the Eatney and Laevis populations that are actually hybridizing to make these Jonesy individuals. And then the lighter colored ones are from a neighboring but somewhat far away population. And you can clearly see that they're not, particularly for Eatney, they're not as closely related. And then you can see in this genomic space that there's this sort of linear gradient that sort of continuously varies from much more like Laevis to much more like Eatney. And so this type of population is actually really well suited to doing the kind of thing we want to do, which is mapping these associations between genomic variants and the phenotype. So to just sort of put everything together, I think where we're at is we're pretty confident that this is actually a case of parallel evolution of this really interesting phenotype. And then because we have this like real-time study system where these things are hybridizing and they're really sampling the genetic and phenotypic space, we're really well suited to go in here and sort of probe which genomic regions might be actually underlying that variation in phenotype. And then we can go back and we can say, well, okay, once we found those, are those the same ones that are in the barbatus virgatus? Because that's actually a much more direct way to get at this question. And I mean, I have no idea. It could be that there's some parallelism at the molecular level. It could be that there are different mechanisms. Really don't know, but that's where I'm going with this now. And yeah, so that's my talk. And I'd like to just acknowledge everybody who I've been working with. So Carrie and her other postdoc, Ben Stone, who's working on sort of a similar problem in a different group of Penstemon. And we and some others have a really helpful discussion group every week where we sort of talk about these statistical approaches. And that's been very helpful for me for learning. And Josh and Hoyt, and Alice, who works on actually collecting most of that data and like setting up this project, and they're out there doing field work, and we're all going out to do more field work in this site in May, and then everybody else in the lab. My wife, Rutenda, who's always like going on a Penstemon hunting adventures with me, and my parents, who I think are watching, who also are constantly finding cool populations of Penstemons. They live down in New Mexico and sending me pictures, so that's places I put on the map to go check out at some point. My previous postdoc advisor, Stacy Smith, sort of really inspired me to focus on plants, coming from the background I did, and opened my eyes to the amazing world of plants. And just another thing that people here might be interested in is that this Professor Boz Wing and I and an undergraduate student that we got are working on a project to try to find a way to directly measure local metabolic adaptation to elevational gradients in a Penstemon species that's native to the Front Range Penstemon. And that I hope has implications for how species can adapt, for example, to climate change and to elevational shifts. And then, of course, I got to thank, the organizers for inviting me, and the Colorado Native Plant Society and the genus Penstemon, for which I made this logo. I like the logo, but it's only because they're plants. So, all right, thanks. Sorry if it was a little long. So yeah, if anybody has any questions, let me know. Zoom got questions.
Yeah, let's go ahead and I'll go ahead and go through some of the chat. And you can go through if you'd like as well.
Yeah, let's see if I can.
Let's see.
Yeah, I'll stop sharing the screen.
Okay. Yeah, I'll go ahead and read off some of these chats whenever you're ready.
Yeah, I'm ready whenever. For some reason, it won't let me stop screen share. Oh, wait, it's because it's on that. On this screen.
Oh,
Probably easier to
okay.
see people not doing that.
Yeah, I think it's back. Yeah, I think it's
Okay, cool.
there we go. Okay, let's see here. Yeah, go ahead and go through the chat here. Yeah, hold on real quick. Okay. It says, we got a question from one of our viewers. Says, do you have any idea how old the Penstemon Etonii laevis hybrid zone? How old that hybrid zone is? I think is maybe the question.
Oh, okay. Like how long has that hybridization been going on?
I think so.
I don't know, actually. I don't think it's probably super long. I mean, the whole genus is maybe like 5 million years old, I think. So, yeah, that's a very good question. We could probably try to estimate that. I mean, this is something that's like going on right now. You can go out and look at it. So,
Okay, it says Andrea she just mentioned that Andrea Wolf and her colleagues presented a phylogeny of for Penstemon in a preprint in BioRx 4. I think is that how you say?
yeah.
Oh, BioArchive, is that
Yeah.
how you say? Okay.
I have read that.
Okay.
Yeah.
And then we have another another question. It says, have you compared the Westinger et al. Tree to the wolf tree? They're generally similar, but some surprising discrepancies. Have you considered these differences? Any ideas about these different results?
I'm pretty new to this Penstemon system, and I haven't thought much about the whole tree for the whole genus. I've mainly been focusing on Habroanthus, and I know Carrie has used RAD-seq before, which is useful for sure. That's another way of sequencing a bunch of genomic data at once. You basically chew up the genome into little bits, and that is also a lot noisier though, and there's a lot of missing data. So this is basically whole genome sequencing, and I think is a much more reliable way to get at the phylogeny. As far as the differences with the wolf tree, I mean they just had so many species in there. And I don't really know what to make of it. It probably just comes down to the methods. But yeah, I mean, I've been focusing on this, and I don't think I could do it a better way with the existing technology that we have.
All righty. Okay, we got here a question from Bob. It says, if Tony, what is hummingbird pollinated and Barbados is bee pollinated, who pollinates the hybrids? Does hybridization negatively impact pollination?
Yeah, that's a great question, and we don't know, and that's one of the things we would like to find out, because if it's been going on for a while, you would think that if there is some sort of maladaptation in pollination for the hybrids, that might show up. I think both Carrie and I have talked about the idea that there might be sort of an asymmetry in preferences as well, because a lot of the bees literally cannot get inside of these long tubular hummingbird pollinated flowers, but hummingbirds can go the other way. And so
Yeah.
like, we're also interested in how the variation is being introduced. Is it mainly being driven by hummingbirds, putting hummingbird species genetic variation into the bee pollinated species, or is it more symmetric? And I would really like to know that. That's a super good question.
Yeah, that would be interesting to know kind of the composition of pollinators. I guess this here, okay, April says great talk. Thank you, Luke. Lisa's happy about our our speakers. We got Pedro here. He says that was great, Luke. Thanks. How far geographically are these hummingbird pollinated species from each other? He says, sorry if I missed that information.
Yeah, no, I didn't present that. And so nowadays anyway, they are, some of them are quite far from each other. So like Labrosis, Penstemon labrosis is in the Sierra Nevadas, California. Penstemon cardinalis is only sort of in southeastern New Mexico and northwestern Texas by like the Guadalupe Mountains. The other two are pretty widely distributed and can overlap. I don't know what it used to look like. And so I think for that, we kind of need to estimate when those events happened and how that matches up with the geographic and, climate changes that have happened since then. But yeah, I mean, maybe it's not too surprising, for example, that cardinalis and Labrosis are independent events, considering they're separated by like 1,500 miles of, deserts and mountains and forests and stuff.
Huh. Yeah, and I think I'm just kind of having a little, I think that's it for from the chat at the moment. If anybody else has any other questions, you can put them in the chat. If you feel free, comfortable, you can also, I think we have enough, we have enough people here that it's oh, we got another message here. So oh yeah, he's yeah, everybody. It's just another thanking you for your for your presentation today, Luke. So yeah, if anybody else has any other questions, feel free to put it in chat, or if you'd like to go and say, I think we have enough people in the present in the audience right now that it's it'll be all right. So do any of you guys have any questions? I think that was great. That was an awesome presentation. I think yeah, that was a little easier for me to understand some of that stuff, because I really don't know too much about all that, and I think you did a really great job of presenting today. So thank you for coming. Yeah, thank you for coming in.
Having me. All right.
Yeah. So I guess oh, we got another message from one more person. Yeah, as far as this being posted in the website, I don't know, you could probably contact Luke, and we did make a recording today, and if you're comfortable, I'm sure you contact him and see about acquiring like a copy.
If it's okay with the Native Plant Society, I can put it on my YouTube channel and just have a link that.
Yeah, it's fine. That's fine with us. It's we usually just make sure it's okay with the presenter and everything.
Okay.
So
Cool.
Cool. Well, thanks for everybody for coming today. That's pretty much it. We kind of went through most of our announcements already a little earlier today. So yeah, the only other thing I could say is, check out the plant. Make sure to check out the what do you call the plant sale today, and we also have like our the plant swap coming up as well. And if you're interested in the field trips, feel free to sign up for those as well. So cool. Well, yeah, thanks everybody. Thanks all everybody online for attending. And yeah, I guess you guys, I hope you have a good evening. All right, cool. That was awesome.